Bismillahirrahmanirrahim. Assalamu alaikum. Once again, I am with the first year biology, HSSC Higher Secondary School, and I welcome you all in my YouTube channel, exploring the concepts once again. And my topic once again is from Bioenergetics, that is chapter number 4. And one thing which is interesting, it is actually the extension of my last lesson, right? So we have discussed light dependent phase and non-cyclic photophosphorylation and the two stages of non-cyclic photophosphorylation in the last lesson. So before starting, I need to tell you what are my objectives today. I want you people to understand the term non-cyclic phosphorylation once again, and the understanding of another important term, chemiosmosis. What is chemiosmosis? Why it is important? It will take place, and how it will take place in the chloroplast. So it's our target today. So before starting the lecture, I need to have a quick review from the last lesson, as it's an extension. So first question is, what is phosphorylation? It is actually the addition of phosphate group to ADP, adenosine to diphosphate, and the result should be the ATP. And that addition of phosphate group to any molecule, especially to ADP, is called as phosphorylation, and the resulting molecule is ATP. So that what is called as phosphorylation. And then next question is, name various steps of non-cyclic photophosphorylation. So this is we are going to discuss once again in this lesson as well, and name the molecules which are responsible for oxidation and reduction of chlorophyll. So you need to tell me the name of the molecules that are responsible for oxidize the chlorophyll molecule and to reduce it. So if you recall the topic, that photons, what are photons? Photons are the energy packets which are in the form of heat. They have no mass and no energy at all, and sorry, no mass and no charge at all. They have energy in the form of heat. They transfer their energy to the electrons of the chlorophyll molecule, and these highly excited and highly energized electrons, they will move out of their shells, and primary electron acceptor of chlorophyll molecule A of PS2, they have to, it has to give their electrons to primary electron acceptor. So primary electron acceptor is responsible to oxidize the chlorophyll molecule. What is oxidation? It is actually the removal of electron, or the removal of hydrogen is called as oxidation. So what we have seen in this reaction, there is a removal of electrons. So chlorophyll get oxidized with the help of primary electron acceptor. First part of the question we have solved, right? And the next part is, who is going to reduce it? Which molecule, I mean. So the water molecule, photolysis. If you can recall the photolysis is responsible for giving out of its electron to the molecule of the chlorophyll, and addition or the gain of electrons and gain of hydrogen is called as reduction. So chlorophyll molecule get reduced with the help of the electrons coming from the water molecule. So at the same time, it is going to reduce and oxidized. So this is a very special, scenario. Find out a very beautiful natural situation. Okay, so that's why photosynthesis is called as redox reaction. So importance of photolysis, how what is photolysis, how it is important to the photosynthesis. So photolysis is actually the breakdown of water molecule in the presence of light with the help of some specific enzymes. So that what is called as photolysis. And how it is important, or why it is important. So it is important because it is going to give the electrons to chlorophyll molecule. It is responsible to make the chlorophyll molecule stable. And the next function is, it is going to help in the replenishment of the oxygen in the atmosphere. Finally, it has given its hydrogen molecules to NADP for the formation of another very high energy compounds, NADPH, which is responsible and which plays its role in Kelvin cycle. So let's discuss the today's topic. non-cyclic photophosphorylation. So we have started this topic and we have completed two stages, and the topic is non-cyclic, right? It means the beginning and the end of the reactions are totally dissimilar, right? And the electrons from PS1 to photosystem 2 will reach to photosystem 1. It will not come back to its own photosystem 2, right? So photophosphorylation is actually the production of phosphate ATPs, the production of ATP, or you can say the addition of phosphate molecule to ADP in the presence of light is called as photophosphorylation. So stages are absorption of light by PS2 and excitation of its electron. We have completed this phase and this stage, and then is next is we have completed this, right? And next is photolysis. We have completed this as well. And what else is the next step, which is highlighted one? Electron flow from PS2 to PS1. It's my today's topic, right? We have discussed, we are going to discuss only electron flow from PS2 to PS1, which is actually the third step. And then is the absorption of light by PS1 and excitation of its electrons. And then fifth point is electron flow from PS1 to NADP. Last two steps, fourth and fifth, we are going to discuss in our next lesson, right? So let's start the electron flow from PS2 to PS1. So first of all, we need to know the two photosystems. Photosystems, you all know, it has a reaction center, it has the chlorophyll molecules, all these green circles. Let me take the pointer. Yes. All these green circles are actually the antenna complexes, and this blue region is actually the reaction center containing the chlorophyll A molecule and the primary electron acceptor. So when light strikes or falls on the antenna complexes, the energy will travel from through these the antenna complexes and reaching up to the chlorophyll A of PS2. Why it is PS2? Because it is discovered after the second one, although PS2 is responsible for the initiation of the process. So still, so I don't know why they're not going to change their numbers. Anyway, so this is PS2 P680. It has a chlorophyll. It has capability to absorb P680 nanometer wavelength. So that's why it is called as P680 photosystem or phytochrome P680. Okay, so whenever the photon strikes this, it is going to leave its electron. It has to leave two electrons as two photons are going to strike it, and these two electrons are taken up by the primary electron acceptor, which is a protein called pheophytin. So whenever it takes in two electrons, obviously it gets reduced, right? So it is not it's a stable state, so it want to give it to the other molecules. So through electron transport chain, there is electron transport chain which is missing in this diagram, as I want to discuss in the next slide. So the electron, these two electrons which are highly energized, right? These highly energized electron, after moving through the electron transport chain, will become the weak electrons, right? They will become the weak electrons, right? And there is no place for them. So what about the photosystem 1? Photosystem 1, obviously it's a photosystem, so it will receive photons once again, and it has two the antenna complexes and the reaction center. So it will once again, it will too strike like the chlorophyll, like the photosystem 2. So it will have to remove two electrons from P700. It has the chlorophyll molecule which is responsible to absorb a wavelength a wavelength equivalent to P700, right? So the electrons coming from the PS2, they are, as there is no place for them. So and for fourth system 1, there is no photolysis takes place to fulfill this place. After removing the electrons from the P700, these two highly energized electrons, obviously there it is impossible for them to retain inside the chlorophyll, so they too have to move out of the photosystem, and there is two vacancies created in the photosystem 1, which is fulfilled by the two weak electrons coming from the PS2. Now let's discuss how these two weak electrons reach up to the PS1. Now, electron flow from PS2 to PS1. Here is a few a text just to tell you. If you want to read through it, you have to go through it before for your complete understanding. So PS2, PS2 is photosystem 2, and PS1 is photosystem 1. PS2 is the electrons from PS2 will move from to PS1 through electron transport chain. Through electron transport chain. Electron transport chain has few proteins. Electron transport. It is a cluster of proteins. It is actually a cluster of proteins which have the capability to move the electrons. That is why it is called as electron transport chain. And it is also responsible because if these highly energized electron, they are not passing through this electron transport chain, their energy will lose. They will lose their energy in the form of heat without producing any productive product, right? So electron transport chain is responsible for the proper utilization of the energy trapping by these electrons from the sunlight. So ETC has few proteins which has some metal ions for easily absorption of electrons. Plastoquinone, the first protein. The first protein of the ETC is called as plastoquinone. That plastoquinone is actually an iron containing protein. Then is cytochrome complex. Cytochrome complex has two iron containing proteins. It's a complex. It means it is not a simple compound. It's a complex compound and it has two proteins. One is cytochrome B, another is cytochrome F. And then the third protein is plastocyanin, PC. Okay? Plastocyanin is actually the plastocyanin, another copper containing protein. So these metallic ion containing proteins are responsible for the movement of electron through ETC. And after moving through these proteins, the energy of the electron will harvest into ATP. This is the non-cyclic photophosphorylation. This is photosystem 2, photon, and photolysis, right? In this diagram, you can see the photolysis. The water is going to take two electrons to P680. P680 is already have get strike through the like photons, and then two electrons are coming out of the P680, and who is going to take it? Primary electron acceptor, which is a protein called pheophytin. That pheophytin is going to drop its protein to plastoquinone. I already have told you it's a iron containing protein. Then these two electrons bond making and bond breaking will going on here. Pheophytin get oxidized and plastoquinone get reduced by taking the electrons coming from primary acceptor. Then plastoquinone will drop these electrons and the the oxidation of the plastoquinone will take place through cytochrome complex. Then the oxidation of cytochrome will take place through the plastocyanin. And after moving through all these proteins, so the bond making and bond breaking obviously will take energy of the electrons. Even then, these two electrons have that much energy that it will generate a molecule of ATP. And then after reaching plastocyanin, plastocyanin will drop these two electrons, and as they are very weak now and they have no energy. And these two electrons, is by the way, are received by P700. These two electrons are received by P700. I already have told you in the last diagram. This is plastoquinone, plastocyanin. So we have completed up to this point. Now, what is the purpose of moving these electrons? As you all see that the production of ATP. What is this production of ATP? How it will possible in this reaction? Let's see. The cytochrome complexes. A very important point about cytochrome complex is that this complex is not only an electron carrier, but also work as proton pumps. It means they can pump the proton by utilizing the energy, right? Whenever you see the word pump, it means some protein is there which is responsible to work, but on expenditure of energy, right? So they are, although they are acting as electron carrier, but along with the electron carrier, they are also working as the proton pumps. So these cytochrome complex, although they are taking the electrons from primary acceptor, so that's why they are called as electron carrier. But along with the carrier, it also have the capability to pump a proton. Okay, now let's discuss this diagram. First point is the electron flow through the cytochrome complex stimulates it to pump the proton from stroma to thylakoid inner space. This is the thylakoid inner membrane. You all can recall that the structure of the chloroplast. Structure of the chloroplast have thylakoid. Thylakoid, the pile of the thylakoid is called as the granum. And the thylakoid membrane, each thylakoid has thylakoid membrane, which is actually a lipid bilayer. And this is the membrane of the thylakoid. This is the thylakoid lumen, and it is the stroma, right? So thylakoid lumen. So what they do, the stroma has the hydrogen ions which are pumped by the cytochrome complexes to the thylakoid lumen once again. So, what is the purpose of doing this? In this, the energy of flowing electron is transformed into a gradient of protons in the thylakoid inner space. This is the purpose. By taking the energy from the electrons, these protons will become high energy protons. They are becoming the high energy protons, right? Just keep this in your mind that the movement of hydrogens is from stroma to thylakoid lumen. Is from stroma to thylakoid lumen, right? Through cytochrome complexes. This movement is energy driven, right? This movement of protons from stroma to the thylakoid lumen is energy driven. Chemiosmosis. Chemiosmosis, right? Chemical process. There is something involved chemicals. And osmosis, movement of water, right? The word osmosis means the movement of water. What is osmosis? Osmosis is actually the movement of molecule from higher concentration to lower concentration, first of all. Secondly, always involves a membrane. This is the difference between osmosis and diffusion. In both the movements, we can see that movement is from higher to lower concentration. But in case of osmosis, we know that there is a use of membrane always. Right? So what is chemiosmosis? Is actually the reaction that involves the flow of electrons. First of all, the flow of electrons, then pumping of protons, and then generation of ATP by involving membranes. Which membrane? Thylakoid membrane is called chemiosmosis. Now look at this diagram. This is actually what is osmosis is. Higher concentration, lower concentration, and then this is the semi-permeable membrane, right? You all know that it involves semi-permeable membrane. This is a stroma, and these are the thylakoid lumen. You can see there are a lot of dots inside the thylakoid lumen. What are these dots? These dots are protons. Protons means hydrogen ions in the form of ions. So all these lumens are filled. It means that hydrogens, by the way, are moving from stroma to the thylakoid, and there is creating a high amount of hydrogen inside the thylakoid. So I'm hoping that at least you have made few points in your mind that the movement of hydrogen is from stroma to lumen. By having this reaction, we have seen that thylakoid lumen are filled with hydrogen, and there is a higher concentration of hydrogens inside the thylakoid membrane. Now what is the purpose of making this concentration high? So next is look at this diagram. All these hydrogen ions are showing you the amount of hydrogen. This is thylakoid lumen and this is stroma. The stroma has few or less number of hydrogens as compared to the thylakoid lumen. How or why? These are in this thylakoid membrane, and sorry, inside thylakoid lumen, we can see that hydrogen water molecules photolysis will take place and hydrogen ions are formed. There are so many hydrogen atoms obviously are formed because there are so many photolysis reactions will take place. So by doing this, what happened? These hydrogen ions, there are accumulation of hydrogen ions, this high hydrogen ions, creating a force which is going to pump the hydrogen from this carrier protein. It is a carrier protein which is called as ATP synthase. This is a protein which is called as ATP synthase. It is a channel protein. It is providing a channel, a pathway for the movement of hydrogens from higher concentration to the lower concentration. Okay, it's a simple movement of hydrogen from higher concentration to lower concentration, as these hydrogens have high energy. From which source they have got the energy? Let's see. These hydrogen ions are moving through this channel protein, and moving through the channel protein, the energy of the hydrogens is utilized for the production of ATP through ADP and PI. And these hydrogens will come to the stroma. This stroma, when will come to the stroma, this hydrogen will, when will be the part of the stroma, these hydrogens are like cytochrome complexes, basically pump these hydrogens through these cytochrome complexes into the thylakoid lumen once again, by taking the energy from the electrons. So these hydrogens have the high energy from the electrons, right? They are taken there in this energy from the electrons. So this is the movement is which is responsible for the production of ATP. I repeat the process of chemiosmosis once again. Chemiosmosis is actually the movement of electron. This from what is the source of electron? Source of electron is PS2. Electrons coming from PS2 going to the quinone, plus to quinone, then to cytochrome complex, then to plus to cyanine, and to finally to the PS1, right? After moving through this ETC, the energy of the electron, they will lose their energy in, first of all, in the movement of the through the ETC. Secondly, they are giving their energy to the hydrogens there, and cytochrome complexes, they utilize the energy of electrons for pumping the hydrogens from stroma to the thylakoid membrane. Okay, first of all, the first point is the movement of the electron through ETC. The second point is the source of hydrogen is also always higher in the thylakoid. Why? Because of the photolysis. Secondly, when this hydrogens, when there is a high accumulation of hydrogen ions inside the thylakoid membrane, they push the hydrogens from higher concentration to the lower concentration through a channel protein. It's a passive movement, right? It does not require any energy. That's why we call it as osmosis, because osmosis is a passive movement from higher concentration to the lower concentration involving a membrane. So hydrogen will move through this channel protein, and the energy stored inside the proton or the hydrogen ion. This energy is utilized by ATP synthase. This knob-like structure is basically the ATP synthase, an enzyme which is responsible for the production of ATP. And this hydrogen will come into the stroma. Once again, cytochrome complexes will take this hydrogen and pump inside the thylakoid lumen. Now, I'm hoping that you will understand that process. It's called as chemiosmosis, the movement of the electron, the movement of the proton pumps, and the production of ATP is called as chemiosmosis. So how is ATP produced? It's a very beautiful diagram showing you the both the reactions, photosynthesis and respiration, and their, the production of ATP. So thylakoid space, basically both of the reactions are reverse of each other. We will discuss this diagram in while we were discussing the mitochondria. Now, phosphorylation chloroplast. Now, read this para carefully. The proton gradient activates an enzyme in thylakoid membrane called ATP synthase, which not only moves the proton back into the stroma, but also catalyzes a reaction in which ADP and PI are combined to form ATP, which is called as phosphorylation. The energized electrons now, after losing their energy, move from cytochrome complexes to plastocyanine and finally incorporated into the PS1. Once again, non-cyclic photophosphorylation, a brief review. So light strikes the chlorophyll molecules, and then to P680 reaction center. Primary electron acceptor will take two electrons. These two electrons will be taken by the primary electron acceptor, and there is a vacancy of two electrons in P680. This vacant space is fulfilled by the electrons coming from the water molecule. It will incorporate into the P680 into the chlorophyll molecule, and the oxygen will go to the atmosphere. The hydrogen will obviously, we will use this hydrogen too in the last of the reaction. Then is the electrons from the primary electron acceptor will move to the plastoquinone. After plastoquinone, it will move to the cytochrome complexes. And then cytochrome complex, it will move to the plastocyanine. By when the electron is moving through this electron transport chain, the energy of the electron will store in the form of ATP. This is called as chemiosmosis. And then finally the last reaction, we will then electron will the coming electrons from the weak electrons from the etc first etc and first and second and ps2 and first etc. The weak electrons coming from ps2 and first etc will incorporate into the primary electron into the p700. Why? Because there is no place and there is no photolysis as well for the ps1. So this is up till now we have completed up to this point. Recap. You need to tell me the answers of the following questions. Name the major proteins which are present in the first ATC. You need to tell all of the name: plastoquinone, cytochrome complex, plastocyanin. Then what is the importance of cytochrome complex in ETC? They are very important. They are not only acting as the electron carrier but also as the electron pump. And then what is ATP synthase? It's a protein present in the thylakoid membrane, and it is responsible for the production of ATP, and it gets activated by the high concentration of protons inside the thylakoid lumen. How would you define chemiosmosis? Chemiosmosis is actually a coupled reaction. It's a very beautiful, in fact, you can say a marvel of the nature, and it is actually that natural products, natural system are itself is capable of producing energy in the form of ATP for itself. Chemiosmosis is actually the movement of electron along with the movement of the proton pumps by creating a proton gradient, and the production of ATP involving the membrane is called as chemiosmosis. Now you need to explain this diagram with the reference of the chemiosmosis. This is your home task. You need to solve this question on your own and write me the answers in the comments. All the best from my side. I'm hoping that this topic, which is called as chemiosmosis, is most of the time students get confused about these questions about this topic. So I'm hoping that this lesson would be helpful for you all. I thank you all, and please don't forget to subscribe my channel and share all of my videos. Thank you so much. Allah Hafiz.